We all know that we have thread-like structures known as chromatin in our nucleus. This chromatin is chemically DNA, deoxyribose nucleic acids. These DNA are the blueprints that code for all our characters or traits. Imagine if there is a change in any part of this DNA, that will change the trait that is coded by that particular part of a DNA. Such changes can therefore be very damaging. These changes in the DNA of a gene are known as mutations. The DNA in our nucleus is present in the form of very long threads that are packed as chromosomes. Packing a chromosome means DNA is coiled and condensed. Now the changes in DNA can also be in the form of changes in chromosome itself. The changes in chromosomes would affect a larger part of DNA. This would result in changes in large number of genes. This is because, as we just saw, chromosome is made up of condensed DNA. So even a small part of chromosome means a long DNA segment. So such changes are therefore known as chromosomal aberrations. Chromosomal aberrations can be deletions or loss of a part of chromosome, insertion or gain of a part of chromosome, and duplication, where again a part of chromosome is present in double the amount or 2 copies. Now let's get back to mutations. Gene mutations involve changes in genes. These are sudden and random. Mostly we do not know the cause of it. These are the very important sources of variations. In the previous unit, we studied as sexual reproduction as the most important cause of variation. Next to sexual reproduction, gene mutations are the next most important sources of variation. For gene mutations to be effective as variation, they need to be inheritable. Inheritable means these mutations should occur in cells which form the gametes, so that these mutations can be passed on to the next generation. They can be spontaneous, that is occurring on their own, or they can be induced, that is made to occur. Now the agents that induce mutations are known as mutagens, chemical agents, alkylating agents such as nitrous acid base analogues, and physical agents such as radiations. Gene mutations are mainly of two types. The first one is the point mutation. Here, one base in the DNA sequence is changed to something else. For example, in the sequence shown here, supposing the G at the third position changes to T, then this is point mutation. The sequence of rest of the DNA remains same, only G is changed. Now, as you will study in the next lesson, 3 sets of bases form one amino acid. So AAG forms amino acid 1, ATC forms amino acid 2, and so on. Now let's say as a result of all these amino acids formed, a protein P is formed. These amino acids join together to form the protein P. So if AAG changes to AAT, instead of amino acid 1, some other amino acid X will be formed. Now since one amino acid is changed and these amino acids are joining to form the protein, so the nature of protein also changes. Protein changes, the pigment or the enzyme it forms will change, and this is how change in one base can alter the trait. The second type of mutation is frame shift mutation. Though we'll be discussing this in more detail in next chapter, a short introduction is essential here. In frame shift mutation, there is deletion or insertion of single base. So either a base is removed or a new base is added. Now let's take a sequence. There are 3 base frames for making amino acids. Now supposing G here is removed. The first 2 frames remain the same. they'll form amino acid 1 and 2, but thereafter the frames will be disturbed. So now instead of amino acid 3, 4, 5 and 6, all new amino acids will be formed because the frame that was including G will have to shift to CCT now. TAA will become AAG, GCC will become CCT, and TAA will have to take another base from the next in sequence, and AA and the new base will be included in the frame. So entire frame has been changed or shifted except for the first two amino acids. Remaining amino acids are different. They are not as before. So the protein that is formed will also change. A new protein will be formed. A similar situation will arise if a new base is inserted at some position, maybe here or any other position. Wherever the new base has been inserted, all the frames after that will change. So now can you understand why these mutations are known as frame shift mutations? Now mutations are very important for evolution since they are a source of variations. But this is only possible if mutations are passed from one generation to next. That is, they are inheritable. When will they be inheritable? When they occur in germ cells or the cells that are involved in gamete formation. But this is one sequence. What if mutation occurs in somatic cells? That is, other cells of the body. They might form a non-functional protein, and the process that involves the altered protein, that will stop. Now this leads to genetic disorders. While some of the genetic disorders may not be very serious, some may make life very difficult. Whether they are easy to handle or not, genetic disorders are extremely difficult to cure. So it is very important that we understand how they are inherited and how they can be managed. The way we study inheritance in P or Drosophila, that is by crossing selected organisms, is not possible in humans. In humans, inheritance of traits is studied by analyzing previous generations and how the trait has been passed on in those generations. This type of analysis is known as pedigree analysis. Here, a trait is studied in ancestral generations as far back as accurate ancestral data is available, that is generally 3 to 4 generations. And on the basis of data obtained for the inheritance of a particular trait, a pedigree chart like this is drawn using certain symbols. And finally, on the basis of these symbols or these charts, the future inheritance of the trait is predicted. Now these are some of the symbols that are used in pedigree analysis. For male, a square is drawn. For a female, a circle is drawn. If the sex is unspecified, then a rhombus can be drawn. Now these shaded square, circle and rhombus mean that the trait that we are studying is visible in these individuals. So if a square shaded is drawn, that means the male is affected. If the circle is shaded, that means this particular female is affected, and same way for rhombus. A single line connecting a male and a female indicates mating, and two parallel lines connecting a circle and a square means that the mating is between relatives, that is consanguineous mating. Now, in this pedigree chart, the parents are shown above and the connected lines show children. And this sequence is from birth. This would be the first born and this would be the second born. So from left to right in the sequence of birth. Now, this particular chart shows that they have a parent which is not affected, but the male child is showing the traits. Sometimes a rhombus with a number is written, which indicates the number of unaffected offspring. So gender is not specified, there is no need to specify, but the number indicates how many offsprings are there, and all of them, since they are unshaded, so they are unaffected. So these are the symbols that basic symbols that we use in pedigree analysis. So now that you know the symbols being used in pedigree charts, you can make some sense of these images. Again, these charts are used for analyzing inheritance patterns of traits and make deductions about the nature of genes controlling these traits. So two of the most basic things that you can deduce on the basis of these charts are whether the genes are dominant or recessive, and whether the gene is autosomal or sex-linked. Now let's see what clues will tell you whether a gene is dominant or recessive. Now, do you recall Mendel's first law, the law of dominance, when you crossed a capital TT with a small t and then you got a heterozygous capital T small t. So in F1, all progeny showed only the tall character, which was the dominant trait. The recessive trait was not visible. But then again in F2, the progeny showed dominant and recessive in the ratio 3:1. So you can see that if you see 3 successive generations, recessive trait is seen in 1, it is missing in 2nd, and then reappears in 3rd. So whenever you are given a pedigree chart, there will be at least 3 generations shown. All you have to do is check whether the trait is visible in all generations or if it skips a generation. If it is visible in all generations, it is a dominant trait, and if it skips a generation, it is a recessive trait. Now you can see this pedigree chart. The trait you are asked to discuss is the one in red. Now first is the parental generation. There is no red. So trait is invisible in parental generation. Then it appears in F1 and is also there in F2. So since this trait skips one generation, that is the parental generation, it is recessive. Now try deciphering whether the trait in these pictures is dominant or recessive. Try solving these. Now how to find out whether the trait is autosomal or sex-linked? For this, you have to recall that in humans, sex chromosomes are X and Y. Now when we say sex-linked genes, the gene can be on X or on Y. Let's leave Y for now and focus only on X. In females, there are 2 copies of the X chromosomes. So genes on X chromosome in females behave just like the genes on autosomes. 2 alleles may be dominant or recessive. In males, the sex chromosomes are X and Y. Here, the Y chromosome is not homologous to X. The genes present on X chromosome do not have a homologous allele on Y chromosome. So it has no corresponding allele. If the corresponding allele is absent, then if the allele present on X chromosome is dominant, it will be expressed. Now even if it is recessive, since it has no corresponding allele on Y, this R will be expressed, or the recessive allele will be expressed even if it is in single copy. Now let's understand this situation. Suppose a trait is recessive and is located on X chromosome. Now, in some females, it can be capital RR, so small r is not present, it is not expressed. If it is capital R, small r, it is present, but since capital R is present to suppress it, small r will not be seen. If it is small r, r, then only in females this trait is expressed. Now, if it is male, if it is a single copy of R on X chromosome, small r is absent, the trait is not there and it is not seen. But if a single copy of small r is present, then since there is no corresponding capital R to suppress it, it will be expressed. So in males, a recessive trait, even if present in single copy on the X chromosome, will be expressed. With this, you have to remember that recessive alleles of X-linked trait is rare. So chances of small rr homozygous condition is even more rare. So chances of female showing an X-linked trait is extremely rare. What does this mean? That in a pedigree chart, an X-linked recessive trait will be more often seen in males but not in females. So, if you see a pedigree chart where all females are either carriers or shown with partially shaded circles like this, this means that the trait is X-linked recessive and the affected ones are only males. Not all males will be affected, but whichever individuals are affected will be represented by squares and would be males. So this means that the trait is X-linked recessive. With this, we come to the end of our session, but before we end, let's make a list of disorders that we need to study and classify them as autosomal or sex-linked, dominant or recessive. For today's session, try collecting as many pedigree charts as you can and try to decipher the nature of the traits. Happy learning. Thank you. Bye-bye.